It's my great pleasure to announce our first speakers, both colleagues and very good friends of mine. So the first is Dr. Sandi Radko-Juettner, who is currently the Research Program Manager for the Hematological Malignancies Program at St. Jude Children's Research. Prior to this, Sandi completed her PhD in Charlie Roberts' lab at St. Jude. And speaking alongside Sandi is Dr. Hong Yu, who completed her PhD in Peking University and then did postdocs in Mark Vidal's lab and the Fisher lab before accepting a position here with us in the Fisher lab as a staff scientist. And I'm going to hand over to you two to tell us about your work.
Great. Thank you so much, Catherine. I know Hong and I are excited to present and discuss our work today, recently published in Nature, which is targeting DCAF5 and suppressing SMARCB1 mutant cancers by stabilizing switch SNF. Just a brief confidentiality disclaimer. So rhabdoid tumors are one type of solid tumor that the Roberts Lab utilizes as a model. And these tumors are multi-lineage, poorly differentiated tumors that occur in children less than 5 years of age. They occur largely in the brain, liver, and kidney, as well as soft tissue. And in respect to treatment modalities, they're largely resistant to chemotherapy and most of the modes of treatment in the clinic. When we look at the somatic mutation frequency across cancers here on the x-axis, what you can appreciate is rhabdoid tumors here on the left have a very quiet genome. And the only sole mutation in rhabdoid tumors is this biallelic inactivation of SMARCB1, which is a subunit of the switch-SNF chromatin remodeling complex illustrated here. In particular, SMARCB1 is present in 2 out of the 3 subcomplexes of switch-SNF, CBAF and PBAF illustrated here, but it is not present in this 3rd subcomplex, NCBAF or GBAF here. What's interesting about switch-SNF and why the Roberts Lab is in particular interested in studying this complex is we found over time that the complex is mutated in greater than 20% of all cancers. And this is illustrated here from a wonderful review from our lab, where you can see on the x-axis the various switch-SNF subunits, and on the y-axis is the various slew of cancers that switch-SNF is mutated in. So let's get a little bit more into what this complex actually does and its function. So switch-SNF is a chromatin remodeling complex, as I've mentioned. It harnesses ATP in order to mobilize nucleosomes and create an open chromatin state for the recruitment of transcription factors to come in and turn on gene expression. More recently, there's been some structural insights from Sigal Kadoch's group showing that the BAF structure in particular forms this C-shaped clamp around the nucleosome illustrated here, and I've marked SMARCB1 in particular since that's the focus of this talk. And SMARCB1, if we look on the right, is really like the thumb holding the ping pong ball in this case, which is the nucleosome, and it facilitates this integral contact for nucleosome binding, specifically at the acidic patch. So work from our lab from a previous postdoc, Xiaofang Wang, has also shown that SMARCB1 loss in rhabdoid cancer leads to reduction of switch-SNF complex components, and this leads to impaired enhancer targeting, which drives oncogenesis. And interestingly, what we have found is when we treat with the proteasomal inhibitor MG132 over time, we can actually rescue the levels of switch-SNF complex components. And what this indicates to us is that in the absence of SMARCB1, switch-SNF is being degraded. And this is really a good segue into the data that Hong and I will be presenting today. So one thing we have to keep in mind is, unlike other cancer subtypes, we cannot target the sole mutation driving this cancer. And this is because SMARCB1 is biallelically inactivated and there's no protein to actually target. So this creates an unprecedented need to target, to identify new targets and proceed with therapeutic intervention. And so this is really the focus of the research that we'll be discussing today, which is really identifying novel targets for these rhabdoid tumor cancers. So to address this, in collaboration with the Broad and MIT, the Roberts Lab at the time when this work started had contributed 14 SMARCB1 mutant cancer cell lines to the Cancer Dependency Map project for near genome wide screening over 21 days. And we asked the question, what genes, when absent, specifically kill these rhabdoid tumor cells? From the screen, DCAF5 emerged as a rhabdoid-specific dependency, illustrated here in red, compared to all other targets. And so really our first approach was trying to validate whether or not DCAF5 was truly required in this cancer. So we began by performing proliferation assays. On the left, we have rhabdoid tumor cell lines that are SMARCB1 mutant, and on the right, we have control cell lines that express SMARCB1. What you can appreciate is when we knock down DCAF5 with an shRNA in the dotted lines, there's a proliferation defect only in the rhabdoid tumor cell lines on the left, and this is not apparent in the control cell lines on the right. We also performed an orthogonal assay with the CAGE team here at St. Jude, where we perform cell fitness assays, where we target DCAF5 with a guide RNA and then look at out-of-frame alleles over time in red. The out-of-frame alleles is what actually contains the DCAF5 knockout. So when we look at two independent rhabdoid tumor cell lines that lack SMARCB1, over time we see the decrease of out-of-frame alleles. This indicates the cells cannot sustain DCAF5 knockout. However, on the right, in our MCF7 control cells that express SMARCB1, we do not see that pressure for out-of-frame alleles.
So what's a DCAF5? DCAF5 is, as its name suggested, DDB1 code for associated factor 5. I believe today in the TBD talk, we don't need to introduce too much what's the E3 ligase. And DCAF5 is a bona fide substrate receptor for coding for RAIN E3 ubiquitin ligase. Based on our preliminary data, if you recall, sensitive complex undergo proteasome degradation in the absence of SMARCB1. So we hypothesize that as E3 ligase, the DCAF5 will recognize the unstable SMARCB1 deficient sensitive complex and facilitating their ubiquitination and subsequent degradations. So this will lead to our two specific aims. So first, determine the molecular mechanism by which DCAF5 mediates the regulation of the SMARCB1 deficient sensitive complex, and then kill the rhabdoid tumor cells. And the second, to establish the functional study of DCAF5 in the chromatin regulations. So first of all, our Fisher lab has a very rich history of working with the DCAF families. By co-expressing DDB1 and DDA1, we successfully purified the DCAF5 in vitro, and we determined the chromatin structures of DCAF5, DDB1, and DDA1 complex at a resolution of 2.6 angstrom. You can appreciate the high resolutions of the chromatin map on the left side, and take a deep look. DCAF5 tightly interact with the DDB1 with a canonical helix, loop helix motif. We turned the structure to the back. DDA1 in the purple is common components associated with COL4 complex. Intercalate the beta string into the DDB1 propeller through several conserved residues, and then system of the DDA1 form alpha helix that bind to the DCAF5 by hydrophobic interactions, which further stabilize the complex. Especially our high-resolution CRIM structure revealed that the DCAF5 contain a canonical, evolutionarily conserved WD40 beta propeller repeat domain. Generally, the more conserved of the domain is, the more crucial its function tends to be. Through the sequence alignment, we observed that the bottom face of the WD40 domain is remarkably conserved, which is the dark blue here, indicating its potential role in substrate recognizations. So with this structure in mind, we began exploring the potential substrates. So first, Sandi asked, does the DCAF5 loss alter levels of sensitive components?
So as Hong mentioned, we started becoming interested in the substrates, and we took a very biased approach. So initially, we utilized a rhabdoid tumor cell line G401 here, and we treated with an shRNA for DCAF5. What you can appreciate is that we saw an increase in several SWTSNF complex components, such as ARID1A, SMARCA4, PBRM1, and SMARCC1. And these are in these two complexes of SWTSNF, BAF and PBAF, that normally contain SMARCB1. However, the system is one where we can inducibly re-express SMARCB1, as you can see here in lanes 3 and 4. And when we do that, we know that the complex reassembles, and therefore we no longer see this upregulation of the SWTSNF complex components. So largely, DCAF5 seems to be altering levels of SWTSNF only in the absence of SMARCB1. We also utilized several control cell lines. I'm just illustrating one here on the right, which is HCT116. They express SMARCB1. And again, when we have knockdown of DCAF5, we no longer see this upregulation of SWTSNF, which occurs in the rhabdoid tumor cells. So next, Hong and I wanted to take more of an unbiased approach and really try to evaluate what the DCAF5 substrates are rather than just looking biasly at SWTSNF complex components. And Hong will delve deeper into this.
So by utilizing the DTAG VHL system, as reported by Benam et al. In 2020, so we observed that FTDP HA tagged DCAF5 could be rapidly degraded within 30 minutes upon treatment of the DTAG V1, which will recruit VHL to degrade the DCAF5. And Sandi also plotted the SWTSNF components consistent with the phenotype observed in the previous DCAF5 genetic knockout studies. So when the DCAF5 is downregulated, only the C-bar and P-bar for components are stabilized, rather than the NC-bar component BRD4 here. So by using this sample, the samples treated for 24 hours, Catherine and all explored the global protein level change unbiasedly in the response to DCAF5 loss. So TMT quantification was used to analyze the DTAG V1 treated samples versus DMSO treated cell lysates. So the loss of a DCAF5 was largely specific to SWTSNF members. So of the top 18 stabilizer protein, 9 was the components of the C-BARF and P-BARF SWTSNF complex, whereas the NC-BARF complex member, such as BRD9, was unaffected. So this is consistent with our Western blot results. And like SMARCC1 as top hits in the whole proteomics analysis has captured our eye. So we tested in vitro ubiquinations of SMARCC1 with DCAF5 as the E3 ligase. To identify the functional units of DCAF5, we tried to try 3 different constructs. DCAF5 477 contained only W40 and or helix loop helix motif, and 601 contain extended regions and full-length DCAF5. The strongest activity comes from the DCAF5 477 construct of confirming that WD40 domain has a functional in the substrate recognition, which is consistent with conservation analysis. And on the other hand, Shoya in our lab has run the AlphaFold predictions, and the prediction of the DCAF5 with the individual SWTSNF subunits suggests that DCAF5 primarily interact with SWTSNF through W40 domain. This is individual subunits. So how about the whole SWTSNF complex, which is the functional unit in the cells? How DCAF5 regulated? So then next we purify the recombinant canonical SWTSNF complex, which also called C-BARF here, and then we proceed the in vitro ubiquinations with the DCAF5 477 construct. And then we plot with multiple specific antibody of the subunits individually. Then some subunits could be ubiquinated by DCAF5. And then we hypothesize that DCAF5 may interact with multiple SWTSNF subunits. To confirm this in vitro data, Jimmy Peng team in Sanju has run this in vivo ubiquitination analysis. The ubiquitination substrate protein could be identified by the diglycine tag following the trypsin digestions. The monoclonal diglyc antibody enriched ubiquitinated peptides, and upon the DKF5 knockdown, the ubiquitination of ARD1A, SMARCA4, SMARC-C1 has been downregulated. So this is actually confirmed that DKF5 may interact or and then degrade multiple SWI/SNF subunits. And then let's take a step back and to see a big picture. Through all the Western blot and also unbiased whole proteomics analysis, we have confirmed that DKF5 loss increased the abundancy of SWI/SNF subunits, and with the in vitro ubiquitination and in vivo ubiquitin omics analysis, we identify multiple SWI/SNF substrates degraded by DKF5 in SMARCB1 in mutant cancers, and this is happened through ubiquitin proteasome system. So what this means for these cancers? Is this the mechanism by which DKF5 loss is lethal? So Sandi will show you more functional explorations.
So yeah, as Hong mentioned, since we identified the substrates of DKF5, presumably if we knock out those substrates, we should be able to reverse the lethal phenotype. And that's what we ended up doing. We performed our cell fitness assays with CAGE again. What you can see is that when we knock out DKF5 on its own over time, we cannot retain those out of frame alleles which contain the DKF5 knockout. However, when we combine guide RNAs against DKF5 along with guides for various SWI/SNF substrates, we can now see at day 21, we no longer see this selection pressure. So knockout of the presumed SWI/SNF substrates actually rescues the DKF5 loss phenotype. And so really this identified DKF5 as a synthetic lethal in SMARCB1 mutant rhabdoid tumors. So when we have the expression of both SMARCB1 and DKF5, the cells are viable. When we have DKF5 expression, but SMARCB1 is absent, this gives rise to our SMARCB1 deficient rhabdoid tumors. We can lose DKF5 expression and have SMARCB1 expressed, and those cells are viable. But it's truly the dual loss of both SMARCB1 and DKF5 that leads to a lethal event. So just to summarize this part briefly, we've shown you that DKF5 is a specific dependency in rhabdoid tumors. We've showed you that SWI/SNF subunits are direct substrates of DKF5. And we've also showed you that the loss of DKF5 in SMARCB1 deficient cancers causes lethality by upregulating these SWI/SNF complex components. So now the second part of our talk is really going to focus on the function of DKF5 and chromatin regulation as we're really understanding the function of SWI/SNF complexes. And one of the first questions we wanted to address was, what is the effect of DKF5 loss on SWI/SNF function? So Hong and I have really showed you that SWI/SNF is upregulated, but what does this mechanistically tell us? So we first began by performing ChIP-seq experiments against several SWI/SNF complex components, ARD1A in fuchsia, SMARC-C1 in blue, and SMARC-A4 in gray. And upon loss of DKF5, which is the second track in each of these heat maps, you can see that we gain ARD1A, SMARC-C1, and SMARC-A4 on chromatin. But this doesn't really address whether or not these subunits have scattered genomic binding, or do they localize to the same regions that the complex would normally function at. And so to address this, we looked at the regions gaining ARD1A and then compared to see whether SMARC-C1 and SMARC-A4 gains were at these same regions. And you can see that, yes, as we gain ARD1A, we're also gaining SMARC-C1 and SMARC-A4 at these same sites. And when we rank based on the gains of SMARC-C1 and SMARC-A4, we see the similar phenotype. So now we wanted to really understand was, are these complexes binding ectopically, or are they binding to their normal sites? So to address this, we have SMARCB1 inducible rhabdoid tumor cell lines where we can turn on SMARCB1 expression. So we performed cut and run for SMARCB1 binding in those cells. You can see here we start gaining expression of SMARCB1 on chromatin. And then we then asked, are these sites the same sites that we are gaining SWI/SNF binding at? When we look at that, that is truly the case. So really, the rescued SWTCHNF subunits are binding on chromatin to canonical sites where SMARCB1 would normally function. So this just brings me back to a previous slide that I had mentioned earlier in the talk, which was work by Xiaofang Wang, where we had shown that SMARCB1 re-expression alters SWTCHNF targeting at typical enhancers. So one of the questions we next wanted to address was, are the regions that are gaining SWTCHNF binding, are they also enhancers similar to SMARCB1 re-expression? So we performed ChIP-seq against 3 histone marks, which are canonical marks of promoters and enhancers, K27 acetyl and K4 monomethyl for enhancers, and K4 trimethyl for promoter regions. And upon DCAF5 loss, we can see that at the regions gaining SWTCHNF, they're also gaining K27 acetyl and K4 monomethyl. And so these are canonical enhancer regions. But what about chromatin accessibility? So we know that SWTCHNF is a chromatin remodeler. Is it actually able to function at this point, or is it just sitting on chromatin? And if you recall, I mentioned that SMARCB1 is this critical contact to the nucleosome, and it's essential for chromatin regulation. However, we do still have in our rhabdoid tumor cells SMARCA4, which is this top part of the SWTCHNF complex, and that is the ATPase subunit. So is this enough to actually help move the chromatin and open up accessibility, or is there perhaps another chromatin regulator at play here? So to address this, we performed ATAC-seq, and we were able to show that upon DCAF5 loss, we are gaining accessibility. So that indicates that the complex is indeed functioning. But again, this does not get at the fact that whether these accessibility changes are actually dependent on SWTCHNF function. And so 2 possibilities arise. The first is that SWTCHNF levels are toxic, but we require another chromatin remodeler to come into play. And that's what's opening up the chromatin at these regions. And the second possibility is that DCAF5 loss actually rescues SWTCHNF function, and the SWTCHNF complex being accumulated can open the chromatin. So to address this, we are able to utilize the ATPase inhibitor BRM014, and that specifically targets BRG1 and BRM. And when we do this, we can see in the control cells, we close the chromatin. And then at the sites that have DCAF5 loss associated, these sites are now closing when we add in the BRM014 inhibitor. And so this tells us really that the accumulated SWTCHNF complexes upon DCAF5 loss are functional and remodel chromatin. So just as an interim summary, what we've showed you is SWTCHNF is accumulating on chromatin at canonical enhancer regions, and this is really rescuing the functionality of the complex when SMARCB1 is absent. So what about transcriptional changes? And so we don't really have time to delve too deep into this, but briefly, we also showed that DCAF5 loss leads to the upregulation of genes being expressed. And so what Hong and I want to show is, what are these genes? And so on the left, we perform gene ontology analysis upon DCAF5 loss, and you can see the genes upregulated are largely activating development and differentiation genes. And when we look at genes activated upon SMARCB1 expression, these are the same pathways being upregulated, mainly development and differentiation. And this implies, again, that DCAF5 loss is really mimicking SMARCB1 re-expression. So just to summarize the second part of the talk, SWTCHNF accumulates on chromatin at these canonical sites, which are enhancer regions. And when we lose DCAF5, we can actually rescue the function and activity of these complexes when we talk about chromatin accessibility and transcription. But now, can we really leverage DCAF5 as a therapeutic target? And so to get at this, we actually leveraged the DTAG cells that Hong had previously mentioned we utilized for proteomics. And we actually performed an in vivo xenograft study where we implanted these cells into the flank of athymic nude mice and then treated with our DTAG V1 compound for 2 courses. What you can appreciate is in the blue with the DTAG V1 compound, we get rapid degradation of DCAF5, and this leads to regression of these tumors in vivo. But again, rhabdoid tumors occur in children less than 5 years of age. So if we are going to consider DCAF5 as a therapeutic target, what is the effect of DCAF5 loss in normal cells in development? So to address this last part, we generated a DCAF5 germline knockout mouse model. And so we targeted exon 3 of the DCAF5 locus with 2 guide RNAs, and this creates a premature stop codon before exon 4. And so we were able to generate these germline knockout mice using CRISPR-Cas9 technology. You can see the validation here by Western blot on the right, as well as a genotyping gel. But what's more interesting to us is that these DCAF5 mice are actually viable. And when we compare these mice to wild type mice on the right, you can actually see that they are largely the same. There's no discernible phenotypic differences. This is very interesting to us from a therapeutic standpoint. Whoops. So just to summarize this last part before we get into the model and the conclusions, we've shown you that DCAF5 is indeed a therapeutically attractive target, and these DCAF5 knockout mice are viable and have been up to around 14 months of age currently.
Yeah, after piecing together all our data, we have come up with this model to explain why the loss of both DCAF5 and SMARCB1 is so deadly for the cancer cells. In the regular rhabdoid tumor cells, and the DCAF5 recognizes several specific components and for ubiquitination and disruption when the SMARCB1 is missing, and this keep the SMARCB complex from working properly. So when the DCAF5 is knocked out, accumulated and as shown as Sandi showed previously, the accumulated SMARCB1 deficient SMARCB complex could restore the chromatin accessibility. This allows the complex to make the chromatin more open, which turn on the genes involved in the cell differentiations. So, and as a result, the cancer cell lose the ability to keep the renewing themselves. So, here, like what we suggested is that without a DCAF5, cancer cell with SMARCB1 deficiency might lose their immortality because their genes expression change, then pushing them to become more like a normal cells that cannot just keep dividing forever. To summarize our whole studies, so DCAF5. We have confirmed that DCAF5 is a novel dependency in the SMARCB1 deficient rhabdoid tumor, and DCAF5 is a quality control factor for SMARCB complex, and its loss restore the function of the SMARCB complex and to reverse the cancer phenotypes, and also the loss of the DCAF5 loss actually rather than give more damage for the cells, actually we restore the functions of the SMARCB complex. And lastly, we have confirmed DCAF5 is a potential therapeutic target for rhabdoid tumors. In looking ahead, we have several exciting avenues to further explore the role of the DCAF5. So firstly, we are currently characterizing the genetic mouse model that lack DCAF5. And secondly, we want to investigate the day job of a DCAF5. We want to know what's the normal biological function of DCAF5 in the healthy cells. And lastly, we plan to utilizing our CRIAM structures to screen for the potential inhibitors, which is actually important for potential therapeutic inventions with rhabdoid tumor treatment. And on the other hand, which the DCAF5 drug discovery is actually is important for this field with expanding E3 legacy library for Proteomics rational designs. And now we already have some preliminary data hits in hand, and the ISAR and validation is ongoing. Now, acknowledgement.
So with that, we have a lot of people to acknowledge in this work. It's been really a cross-institutional effort with many members of the Roberts Lab and Fisher Labs. Specifically, we don't have time to call out everyone, but all the people bolded have really greatly contributed to this work. We also have to acknowledge many of the core resources, both at St. Jude, Dana-Farber, Harvard, as well as some of our collaborative labs, the Stewart and Pang and Gray labs, respectively. And of course, none of this work would have been possible without our amazing funding sources listed here. With that, Hong and I would be happy to take questions.